Hello students, welcome back to the channel. In this video, we are going to study about one receptor which is used in cell signaling and one common receptor which is generally used by many signaling molecules is G protein coupled receptor. And we call that receptor as G protein coupled because it uses G protein. And what is a G protein? It is a protein which is binding with energy rich molecule either GTP or GDP. So this receptor is actually a transmembrane protein and it is working with the help of G protein. Many signaling molecules like your epinephrine, your yeast emitting factors and neurotransmitters, they are using GPCR. And nearly more than 10,000 GPCR in human are being used. And they are having one common structure. All of the GPCR are having one common structure means they are having a secondary structure made up of only one polypeptide and it is having transmembrane 7 helix. Check carefully. So this line is important that they are large family of eukaryotic receptor protein. They are having secondary structure in which one polypeptide has 7 transmembrane helix. Loops can be there. You can see specific loops are present and one loop is providing a binding site for signal molecules towards the outside medium and another is having the binding site for the G protein. So this is a structure of receptor that is GPCR. Now, they are generally used in different functions including the roles of embryonic development and sensory reception also. And in human, if we see vision, smell, taste all depend upon GPCR. Structure is similar, G protein structure is similar, GPCR is also similar. What does it suggest? That this protein and the receptors they have evolved very early among eukaryotes. So it means it indicates common ancestry also and it indicates it has evolved very early. Now, if suppose any problem is associated with G protein, it is a reason for many diseases. For example, the disease which causes cholera, your whooping cough, your botulism, and all these diseases which are generally people are suffering, it is because the toxin is interfering with G protein function. And now even pharmacologists have realized that 60% of medicines are exerting their effect by influencing this G protein pathway. Now let us understand through diagrams. See, this is your GPCR. This is G protein coupled receptor. We are using this is a site where the signal molecule will bind. And this is a site where G protein will bind. So first of all, try to see G protein. This is G protein. It is loosely attached and it is present towards the cytoplasmic side. Clear? And it functions as a switch on and off. When GDP is attached, see in this diagram, when GDP is attached, it is actually inactive. The time G protein is inactive. Clear? Now see next. When receptor signal molecule is binding to the receptor, you can see here this is a signal molecule binding to the receptor, then what will happen? The receptor is activated. Now this receptor activated will change the shape and now on the cytoplasmic side it will bind to GTP, inactive G protein. Sorry, in the cytoplasmic side it will bind to inactive G protein. Towards the cytoplasmic side it will bind with inactive G protein and making the GTP to enter and GDP out. Understand through diagram, please understand GTP will enter and GDP is out. GTP is entering and GDP is coming out because of which the G protein becomes active. Clear? So first this much is clear. Now see next. Now this activated G protein immediately separate from the receptor, diffuses along the membrane because it is loosely attached, it can move and it binds to the enzyme. changing the enzyme shape and as well as activity. And once the enzyme is activated, it will trigger the next step, which will lead finally to the response. Okay. And change in the shape of the enzyme or change in the shape of the G protein is actually temporary. Why? Because G protein function as GTPase enzyme also. It will do the hydrolysis of GTP into GDP and phosphate, inorganic phosphate. So after this, once the GDP is again coming back, and you can say GTP is hydrolyzed and GDP is left, then G protein again leave the enzyme because it is now inactive and it will come back to its original state. So again the cycle can be started. This is the way how G protein coupled receptor work. Just quick summary. Remember, whenever G protein are binding with GDP, it is always inactive. And when G protein are binding with GTP, when they are binding with GTP, it is active. And this is possible only when signal molecule binds to the receptor. And after the G protein becomes active, it will activate an enzyme. And once the enzyme activity is started, now it will again come back to its original by doing the hydrolysis of GTP into GDP and inorganic phosphate. So that the G protein comes back to normal state and even enzyme also comes back to the normal state. Is it clear? Means original state, so that again the cycle can start. I hope so this video might be helpful for you to understand the concept properly. Let me know if anything else, if you want, please message me. I'll try to make the videos so that it will be helpful for you. Thanks for watching.